that can influence people's exposure to mosquito-borne disease. So this can be things like quality of housing, how pervious this housing is to mosquitoes, how much water storage occurs around the home, and people's access to public health care interventions. These things integrate to influence where mosquitoes are in the environment, their densities, our contact rates with mosquitoes, and then transmission of mosquito-borne disease, and then these things scale up to influence the global distribution of disease. Now let's layer on top of this climate change and land use change. So urban environments are the fastest growing landscapes on the planet today, and they're experiencing intensifying heat island effects with climate warming and heat extremes. And this is really changing the landscape across which mosquito-borne disease is occurring. It's causing emergence of explosive outbreaks of arboviruses like dengue and chikungunya and tropical megacities that combine high human population densities with conducive tropical conditions for disease transmission. And it's also leading to the invasion of mosquitoes into new areas. So for example, Anopheles stevensii, which is the urban malaria vector in India and South Asia, is now expanding in urban environments in Africa and really challenging malaria elimination efforts now and moving forward into the future. Okay, so most of the work that's been done to date on mosquito-borne disease has focused on the effects of temperature, and again, this is because people have been really interested in how climate change and climate warming is going to influence mosquito-borne disease transmission. And also because mosquitoes are small and they're cold-blooded organisms, so temperature really profoundly influences their metabolic rates. It influences their developmental schedules and their life cycles, life history, and fitness. And it also influences the development rate of pathogens within them. So there's been a lot of work in this area. And so what studies have shown, basically there's been a lot of laboratory work done where they take mosquitoes and they put them in across different constant temperatures and they characterize the performance of different traits and different parts of their life cycle that are important for transmission. And so when we do these types of experiments, we can see that temperature has impacts on traits that govern mosquito distributions and how populations change across seasons and across different geographic areas, and that these relationships are nonlinear. Okay, so for example, we have lifespan, temperature has significant impacts on lifespan, on how quickly they develop from juvenile mosquitoes to adult mosquitoes, on how well they survive in the juvenile environment, and then how many eggs females produce. We can also see that temperature has effects on traits that govern our exposure or our contact rates. So this would be the daily biting rate of mosquitoes. And then you can see that for pathogens developing within these mosquitoes, because they're also experiencing temperature through the mosquito, that you also have nonlinear effects of temperature on things like how quickly mosquitoes become infectious, so the pathogen development rate, and then the proportion of mosquitoes that go on to become infectious or vector competence. So looking across all of these traits, you can see that temperature has these nonlinear effects on trait performance. So you have cool and warm temperatures that limit trait performance, and then you have an intermediate temperature that optimizes trait performance. The other thing I want you to notice is that these traits can have symmetric relationships, or they can have asymmetric relationships, and the temperatures which bound trait performance can vary. And so what's been done is you can fit statistical relationships to these, how temperature and these traits change. And then you can use mathematical expressions like this. This is the basic reproductive number for a vector-borne disease, or more complex models. And you can build these relationships into these models to look at how temperature is going to influence transmission. And so, for example, for malaria, what you can see here is when you do this, when you incorporate temperature dependence into this basic malaria model, you can see that temperature at 25 degrees C is predicted to optimize malaria transmission. And then with these models, we can do things like predict the months of suitability of human malaria transmission by Anopheles stevensii, which is the South Asian vector and is invading Africa, or Anopheles gambiae, which is a native vector to Africa. And what you can see here is you have months of suitability with warmer colors corresponding to more months of suitability. You can see that actually malaria transmission by Anopheles stevensii is predicted to have higher suitability in Africa than the native Anopheles gambiae mosquito. And then you can also use these models to look forward into the future. So if we're going to anticipate how climate change, for example, might influence malaria transmission by Anopheles stevensii across the globe or in India or in Africa, you can see in general, even under severe warming scenarios, which would be the bottom panel there, even though you do see a contraction in overall suitability, so you have less red across those maps, you still have many areas of the world that are highly suitable for transmission of malaria by Anopheles stevensii. Okay, so I'm going to talk to you about the urban malaria Anopheles stevensii system in South Asia, and in particular in India. And so Anopheles stevensii live in highly urban environments, as pictured here. And adults will live and feed largely indoors around cattle or in mixed dwellings. And they'll rest indoors after taking a blood meal. And then they lay their eggs, and their juvenile mosquitoes develop in artificial water containers or storage containers around the home. So these can include 50-gallon drums, underground tanks, overhead tanks, and buckets, et cetera. And you can also see them developing in construction sites and pools of water. So previous work in this system by one of my collaborators, Mercedes Pascual, has shown that, temperature is important, but there are other environmental variables that are important as well. So what she's noticed working in Surat and Ahmedabad is that we have strong effects of relative humidity on this system. So for example, as relative humidity goes up, we see that the number of cases within a given year also increase. So we have these positive relationships with malaria. And when you build a semi-mechanistic model that has relative humidity in there as a primary climate driver, you can see that over time, the model does a fairly good job predicting the interannual variability in malaria cases. So this is for Ahmedabad. You've got malaria cases in black. You've got the data that the model was trained on in purple, and then you have the green. These are the out-of-fit predictions for how the model performs on data it was not trained on. And you can see in general that the model does a very good job at predicting when epidemics occur as well as the magnitude of those epidemics. And what's interesting is we don't know as much as we do for humidity as we do for temperature. So while there has been work characterizing the effect of humidity on the mosquito life cycle and the transmission process, it's not quite at the same resolution as we have for temperature. So it makes it hard for us to incorporate it into mechanistic models to make similar predictions, currently and into the future with climate change. And so this sets the framework for the work that we're doing in India. So this is an NIAID R01 grant, where we're really focusing on redefining our understanding of the climate drivers of urban malaria in the context of relative humidity. And so, for example, in the first part of this project, it's going to involve, and I'm going to talk to you a lot about this today, laboratory work where we're trying to redefine some of those those temperature trait relationships in the context of relative humidity variation. We then use these data to parameterize temporal models that will go in and see if we can do a better job at predicting the interannual variation in malaria cases. And then we're also building spatial models within two cities, Surat and Ahmedabad, to try to identify hotspots for malaria transmission. And so this project is a very large project. It involves 7 institutions. So there are 4 US-based institutions. So Cornell University is the prime, but we're partnering with Mercedes Pascual at New York University, Mike Wimberly at the University of Oklahoma, and then Arindam Nandi and Deepshika Batheja at One Health Trust. In country, we have 3 organizations that we're working with. So we're partnering with the National Institute for Malaria Research. The subrecipient PI is Dr. Rajendra Baharia. And then we're partnering, within the cities with the local municipal corporations. So we're working with the Ahmedabad Municipal Corporation, in particular Raj Sharma, who's the head entomologist. And then we're working with the Surat Municipal Corporation with Kashav Vaishnav. And the whole purpose of this project is to try to build models that the municipal corporations can use to inform surveillance and control to help them achieve malaria elimination in the cities. Okay, so I'm going to present to you a lot of the work that we're doing in my group currently. So where we've gone in and recharacterized the temperature trait relationships that are important for transmission in the context of relative humidity. So for example, we've done, we've essentially taken, juvenile Anopheles stevensii. We've placed them in different trays to let them develop. We've placed these trays across 8 to 9 different constant temperatures and 5 relative humidities. And then we essentially just measure the number of mosquitoes that emerge on a daily basis. And then we also do some other quantification of body size, et cetera. And we use this information to understand how the development rate as well as their survival in the larval environment changes with temperature as well as relative humidity. And so what we found actually is that decreases in relative humidity shifted thermal performance of these two traits toward warmer temperatures. So for example, we have the mosquito development rate here. Again, this is how quickly they develop in the juvenile environment. And you can see as relative humidity goes down, so we go from dark blue to light blue, that overall the thermal performance curve shifts to warmer temperatures. So we're seeing shifts in the thermal minimum of about 7 degrees C, which is massive. And we're seeing a shift in the thermal maximum by 2 degrees and some shift in the thermal optimum. So what this means is that mosquitoes developing in dry environments can develop at much warmer temperatures than mosquitoes developing in humid environments. And then we also see similar shifts in their survival in the juvenile environment. And here we're seeing more shifts in the thermal maximum. So again, we're seeing about 7.5 degrees C shift. So to give you a context of how big these shifts play out over space, those maps that I showed you in the beginning, for stevensii and Gamby, for example, we're talking about maybe a 1.5 to 2 degree difference in the thermal maximum and the thermal minimum for those two species. And we're talking here, this is a 7 degree shift with variation in relative humidity. So this is going to play out across space greatly. And then if you look at how this affects body size, you can see that we're also seeing effects of relative humidity on the temperature effects of body size. So in general, as temperature warms, mosquitoes develop and emerge more quickly from the larval environment, and that means they come out smaller. And so in general, you can see that trend playing out. But if you look at how relative humidity modifies that trend, as relative humidity goes down, so here we've got more red colors corresponding to decreases in relative humidity, the effect of temperature dampens. And this is important because body size is a really important predictor of how many eggs a female is going to produce as an adult, also her survival and her ability to transmit pathogens. Okay, so we did similar experiments in the adult phase as well. So again, here, instead of following larvae, we're following adults, and we follow individual adults. So each of these cups contains one female. We give her blood every day, and we keep track of how often she feeds. And then we also have an oviposition site in there daily so that we can keep track of how often she lays and how many eggs she lays. And then we use this information to estimate lifetime fecundity, her lifespan, as well as how often she feeds on a daily basis. And so here we're seeing more subtle effects of relative humidity on these adult traits. So this is for mosquito lifespan. And so here, what we're seeing is because as temperatures warm, mosquitoes tend to desiccate more quickly. So humid environments at warm temperatures here buffer females. So they can exist at higher temperatures in more humid environments. So the T max is shifting up by about 3.2 degrees C. We're not seeing a shift in the thermal minimum, and then the thermal optimum is shifting a little bit to the right as well, with increases in humidity. For egg production, we're seeing a different effect. So here we have decreases in humidity actually resulting in a higher production of eggs on a daily basis as temperatures warm. And I think what's going on here is that females are sensing that the environment is stressful and they're front-loading their egg production earlier in life, and that's resulting in more eggs being produced earlier. And then for biting rate, we're not seeing an effect of relative humidity. And this might be because we're basically slapping them in the face with a blood meal every day. They don't have to work for their blood meals. And if they actually had a host, like forage in the environment for a host, then changes in relative humidity might affect their feeding behavior. But what we can say here is that changes in relative humidity is not affecting their intrinsic desire to feed or the effect of temperature on that. Okay, so what we can do with these data is we can now put them into a mathematical model that is dependent on temperature and relative humidity. And so this is a mathematical model that predicts, that predicts, sorry, I lost my screen up there, that predicts the number of Anopheles-Stevens-I or Anopheles-Stevens-I densities, okay? And so here we've got lifetime fecundity, that's B. We've got, their probability of surviving the juvenile environment, their development rate, and then their lifespan as adults. And we can use this to predict how suitability might vary for Anopheles-Stevens-I populations. And we can do this in India, and we can also do this in Africa, which is the zone of invasion. And so what we've done here is we've looked at the effect of temperature, and then we've produced maps where we also account for variation in relative humidity. So temperature is like business as usual, and then incorporating relative humidity is what we're doing and is the next step. And so what you can see here is that, in general, when we incorporate the effects of temperature, we have much higher predicted months of suitability across both India and Africa than when we account for the effects of relative humidity. So we have less red colors when we account for relative humidity than not, and that's probably because of the complex of ways relative humidity is affecting the mosquito life cycle. Okay, and then for the parasite traits, we're still doing these experiments, and it's early days, and I can't tell you exactly how relative humidity is modifying the temperature effects on parasites, but that will be coming out hopefully in the next year. Okay, so the bottom lines, from this part of the work is, decreases in relative humidity are having pretty big effects on different traits that are important for mosquito population dynamics. So in the juvenile environment, we saw that decreases in relative humidity shifted thermal performance 2 to 8 degrees C toward warmer environments, depending on the trait in question. And then now the question that we're working on is, okay, why is that? Is it because relative humidity is changing something intrinsic to the larval environment, like surface temperature or some other intrinsic property? Then we also saw that decreases in relative humidity had mixed effects on the performance of adult traits. So this resulted in reduced longevity or boosted fecundity at warmer temperatures, and we're working on metrics of parasite fitness. And then finally, these effects are fairly large. So variation in relative humidity is going to have a significant effect on thermal performance and mosquito fitness, their population dynamics, and because these traits are important for transmission, I'm anticipating pathogen transmission as well. Okay, so now we're going to step out of the lab and move into the field. And I'm going to share with you some of the work we're doing in Indian cities. So, if we're going to move into the field, we need to start thinking about the climate that mosquitoes actually experience. And this happens at a fine spatial scale because mosquitoes live in and around your home and in your backyard. And so variation in land use and land cover is going to influence the climate that mosquitoes actually experience. And so, your backyard could look like this slum in one of the cities that I work in, or it can look like this, village in Africa that consists mostly of mud and thatch houses, or this more south rural landscape that you might have. And so, in order to characterize the climate and how land use and land cover influences, the climates mosquitoes experience as well as their contact rates with people, we need to start collecting data at those spatial resolutions. Okay, so what we've done is we've gone out in Surat and Ahmedabad and we've set up these data logger arrays, so where we've paired indoor and outdoor loggers to keep track of temperature and relative humidity. And so this is an example of what our data logger arrays look like across our sites. So we have paired indoor and outdoor loggers because mosquitoes have both indoor and outdoor phases associated with their life cycles. And then, in Ahmedabad, we have 96 sensors across 48 households. And then in Surat, we have 90 sensors across 48 households. And then we focused on human dwellings, cattle sheds, and then mixed dwellings because this is where we see and collect, a lot of the mosquitoes that we work with. And then the sites were also spread out across socioeconomic variation in the city, and then we tried to get good coverage across all the zones in the city as well. Okay, and so from this, what we can do, is that my time? Okay, so from this, what we can do is we can fit statistical models. Does that mean I have to, am I done? Okay, I'll finish up. I'm almost done. So what we can do is we can fit statistical models to the microclimate data that we're collecting with macroclimate data that we can get from weather stations, as well as satellite data on land cover and land use. And we can use these models to make microclimate maps of both cities at fine spatial resolutions that are important for transmission and control. And then what we can do from this is we can, use our models that we're building that have temperature and relative humidity incorporated into them with these microclimate maps. And now we can make environmental suitability maps of the city for transmission. Now, what we've also done to move from suitability to risk is we've done a socioeconomic survey in both of these cities. We've gone out across 2,000 different households, and we've looked at how wealth, education, access to public health care, et cetera, water storage containers might influence people's risk of acquiring a mosquito-borne disease. And then we're going to layer these two data sets on top to make risk maps of these two cities. And we're going to validate this with data that the municipal corporations are already collecting on mosquitoes and cases in the city. Okay, so I'm just going to show you some of the work that we've done so far. So these are microclimate profiles that we're pulling from indoor and outdoor homes. So we've got indoor environments in red and outdoor environments in green, and then we've got them across two different, we've got temperature and relative humidity across two different seasons, May and August. So in general, you can see that across the day, indoor environments are more stable than outdoor environments. And also indoor environments tend to be a little bit warmer than outdoor environments on average. And then because we don't have all of the relative humidity work done, I'm going to just show you what we can do with temperature alone. So what we can do is we can take these temperature profiles and we can use them with temperature-dependent R0 models, like I showed you previously, for both malaria, and also we can do this for dengue. And we can predict how environmental suitability is going to vary across these two cities. So this is for Surat, and so we don't have interpolated spatial risk maps yet, but these are what you'd predict for each of those logger locations. And then we've got indoor environments on top and outdoor environments below, and then we have malaria in green and dengue in purple. And then the size of the circle and the color of the circle corresponds to increased thermal suitability. And so what you can see here is that, across the different seasons, the pre-monsoon, monsoon, and post-monsoon season, we're seeing differences in size. So thermal suitability is changing seasonally. If you look within a season and across the city, you can see that different point locations have different predicted thermal suitability, and that's because of variation in land use and land cover. And then the other thing you can see is where we have predicted thermal suitability to be high in one location can change from season to season. So seasons changing the relationship between land use and thermal suitability as well across the city. And then you can also see some differences for dengue and malaria in general. So the monsoon season, we get more thermal suitability in both indoor and outdoor environments for both pathogens. But then the post-monsoon season, it almost seems like that thermal suitability stays high for dengue, but starts going down for malaria again. Okay, and then the other thing to consider is that microclimate differences between indoor and outdoor environments could have implications for different components of the mosquito life cycle. Okay, so I'm going to wrap up now. Okay, so the key conclusions from our work are, indoor and outdoor environments are different, and this can result in different implications for predicting environmental suitability and risk for malaria transmission. We saw that the highest thermal suitability was associated with environments and times of season, with cooler temperatures that are closer to the thermal optimum for both of these pathogens. The spatial patterns of thermal suitability shift seasonally with land use. And then the future extensions of where we're going with this work is incorporating the effects of other key environmental variables like relative humidity into these maps. We're going to consider spatial variation and socio-ecological factors that shape exposure so we can get at risk. And then we're going to create interpolated risk maps that the municipal corporations can use to guide future surveillance and control. And so the whole idea here is we build data-driven models that we can understand the transmission process so we can make informed policy suggestions for surveillance and control, and then we can project into the future how climate change and future urbanization scenarios will impact these two pathogens. And yeah, that's it. So these are my collaborators, my funding sources, and I have to really give a big shout out for the insect collectors at both of these municipal corporations who are actually doing a lot of the hard work, especially associated with aim 3 of this project.
Thank you very much.
It was a very insightful presentation. Actually, we are working in Sonbhat district for malaria. So as an entomologist, what I have seen there are cases of malaria, and in the winter season when we were there, since in the winters we have the vector Anopheles fluviatilis. It's a forest and hilly area. My question is that even in the winters, the cases are coming because of this vector, but we are not able to find the vectors. We might thought that the migration of the population will be there, and they must be getting the infection from some other source, since they are tribal people and working, coming from a very poor background. But for our surprise, they are even not migrating from, from one place to another. And we have also asked questions like if they are, exposed to the mosquito bites, or what is it, even we find the vector density is nearly 0 there, but still the cases are coming. So how come we come to some conclusion? And we have also tried some other things for collecting the mosquitoes. It was not there. And another small question is the life history traits, as you have mentioned, are directly and indirectly correlated with the relative humidity and temperature. So for me, I am very astonished to see that the temperature, if it is even high by 2 degrees, it is very detrimental to the mosquitoes and even to ectothermal, organisms. How come then mosquitoes are still expanding and, doing such a havoc in any diseases transmission? So
okay, all right. I'll try to. There was a lot. Is it Vivax or Falciparum? It's Falciparum. Okay. That's a good question. Do you have other mosquitoes that could serve as vectors? Okay. So do you think that could be potentially important? Yeah. Yeah. No, I mean, I agree. So So I'm not familiar with your situation, but what we're seeing in Surat and Ahmedabad is we have these refugia through kind of more hostile seasons, either the winter season when it's dry or the post-monsoon season when it's really, really warm and dry, and with construction. So construction sites are really important, and we have interesting migration coming from rural areas that are endemic into the city, bringing disease, and then vice versa. But it doesn't sound like that's what you have in your area, because they're not migrating. Well, I think that's an interesting question then to follow up on. Yeah, and then in terms of the second question, what was it? The second question was the temperature, right? So again, well, so it's a nonlinear effect, right? So that means, in certain areas of the world that are maybe at cool temperatures that are warming, they may become more suitable, and mosquitoes might be able to invade, and mosquito populations might expand. But if you're already in the permissive area or on the warm end, then you could have contractions, right? And that's what your models would predict. The one thing that these models don't take into account for is mosquito behavior. And anybody who works in the field knows that mosquitoes are active only at certain times of the day, and that might shift depending on the season, and they might be seeking, refugia. So I think that's something that's very important. That's much harder to incorporate into a model, but we're going to start trying to think about that in our future work, especially with construction sites. Because I think construction sites are in potential, at least in these cities, refugia during the pre-monsoon season when it's really, really hot and dry. That's where we find the mosquitoes and people as well.
Thank you.
With infection.
Dr. Santabul, sir.
I just have one question.
Sorry.
First of all, malaria was the predominant mosquito-borne disease, and now we are seeing a shift to dengue. So how does it, your model reflect to that?
Yeah, so actually it's interesting because what we're seeing is that with control. So you're having kind of a positive effect of ongoing control, and climate change, because it's becoming more hostile, is making the control effort more efficient or effective.
So it is less more hostile to vector or to parasite?
Both.
Both. Okay.
Yeah, because the parasite develops within the vector, right? And it's also for the vector. So I think that's predominantly why malaria is on the way out and dengue might be on the way up. And so we're actually writing a grant right now to explore the question, to try to understand the different climate drivers for malaria and dengue and how they interact with control. We're writing that for the Wellcome Climate Impact Award. So we'll see. But like we're very much thinking along those lines.
Good luck on this new
Yeah.
brand.
Thank you, Prabhakaran sir.
Is on brackish water, dirty brackish water, because if you
Sorry.
look at brackish, dirty water,
Okay.
if you look at dengue virus, they breed in relatively clean water. So how does that model apply to Aedes aegypti, for example, because you are looking at Anopheles here? That's one. Second thing is maybe we should talk offline, because my colleagues have developed a heat map to identify from where these epidemics originate, and it's been done for the city of Guwahati and Lucknow, and maybe we should exchange notes later.
Okay, yeah, that'd be great. So in terms of brackish and clean, so I would say in terms of like trying to understand how it might, what we're doing in the Anopheles-Stevens-eye system, I think could translate to the Aedes aegypti dengue system very well, mainly because they share a lot of the same habitats. Now, when we're talking about some of the native vectors in Africa, they're developing in like irrigation sites and, like puddles of water on the side of the road, et cetera. Stevens eye is not necessarily found in those habitats. I would imagine that when we're thinking about how relative humidity, if it's affecting surface tension, et cetera, in the juvenile environment, that solutes and concentrates in those environments will influence that relationship. So it might not, what we're doing in Stevens eye might not extrapolate maybe to like the, other Anopheles species in Africa, for example.
Thank you.
Okay.
One more question from the students, or yes, last question, please.
The more affinity, like there is more affinity for the mosquito to go towards O negative blood. Have you considered that blood grouping and typing in your model? If yes, why? If no, why?
No, we have not.